Protein synthesis is a two-step process that copies DNA in order to create proteins for the cell to use. It is important to note that DNA, or deoxyribonucleic acid, is a double-stranded molecule that contains 4 different nucleotides: adenine, blue; thymine, gold; guanine, red; cytosine, purple. Each nucleotide has a base pair, A to T and G to C.
In the process of transcription, helicase unzips the DNA into 2 separate strands. The transcription complex positions the RNA polymers at the active site for transcription, and both strands enter RNA polymers. Only a specific portion of DNA or a gene is copied. This enzyme matches one of the strands to a complementary strand of messenger RNA, or mRNA for short. This is represented by the dark blue line. RNA also has a unique nucleotide called uracil, which takes the place of thymine.
To begin, the RNA strand enters the TATA box, which initiates the process called initiation. Next, the strand goes through RNA polymerase, which matches complementary nucleotides from the DNA strand. This is called elongation. Finally, the mRNA matches the final codon and leaves the mRNA polymerase. This is called termination.
After transcription, the mRNA goes through some modification that removes unnecessary components and adds a methylated cap and a poly-A tail, pink.
Translation begins with the mRNA entering the ribosome, which is made of RNA, which is composed into 2 parts, the large and small subunit. The first codon or 3 nucleotide base pair to enter is called a start codon. Then the transport RNA, or tRNA, comes and matches with a codon using a complementary anticodon. This tRNA is carrying an amino acid by using a aminoacyl, which connects the tRNA to the amino acid. This amino acid begins the polypeptide chain, which becomes a protein when completed. This whole process is the initiation stage, and this occurs at the E site.
The mRNA continues to enter the ribosome, and the appropriate tRNA match with the correct codons. This is called elongation. During this stage, amino acids continue to add to the polypeptide chain using peptidyl transferase. This is also known as the P site. This continues until an end codon is reached.
When the end codon or release factor is reached, the final stage or termination begins. This results in the mRNA leaving the ribosome, and the polypeptide chain is cut off from the ribosome to become a protein.